Bismillahirrahmanirrahim. In today's lecture, we will discuss about the factor affecting passive drug absorption और active drug absorption का जो वो principle है that has already been discussed. और उसमें active drug absorption में जो I mean के Michaelis Menten equation का रोल है, low concentration पे first order को जो follow करता है, at little higher concentration it follows the Michaelis Menten principle and then going to zero order kinetics. So that has already been covered in the previous lectures, and today's lecture we will discuss about the those factors that affect the passive transfer of drug from the site of administration to the blood. और passive drug absorption में जो important factor है, that is the unionized form of drug that is basically absorbed. So the important law that basically is associated with the transfer of drug involving the passive transport is the Fick's law of diffusion, and according to this law the movement of drug occurs from the higher concentration to the lower concentration across the membrane. So simply means that when you take the drug orally or you are taking the drug by intramuscular or any extravascular route, the movement of drug involving the passive transport is always from the higher concentration to the lower concentration. And since it is from the higher concentration to the lower concentration, there is no need of energy because it is passive, और passive की definition भी यही है कि there is no need of energy, and it is basically driven by the concentration gradient, which simply means that the movement of drug is from the higher concentration to the lower concentration, and it is basically represented by dc over dt, which means the rate of change of drug concentration in the blood is increasing with it, so that's why there is positive sign, and it is equal to k c git and c blood, which means that this is the constant that is called as the permeability coefficient, and it is equal to km over f, which means that this is the partition coefficient between the membrane and the fluid, or between the oil and the water phase, and a is the area of the membrane, d is the thickness of a is the area of the membrane, and d is the diffusion coefficient, and h is the thickness of the membrane. So basically you can say that the movement of drug or the rate of change of drug concentration inside the blood is increasing when you administer the drug. That's why with the positive sign, and k is the permeability coefficient, and this is the concentration of drug in the GIT. So greater is the concentration in the GIT, greater will be the movement of drug from the GIT into the blood, and lower is the concentration of drug in the blood, more movement will occur. So that's why that this is a k into c, that is the concentration of drug inside the GIT minus concentration in the blood, and this again, this is equal to the permeability coefficient, and it is equal to the km over km, that means the membrane, f means the fluid, that is in this case the fluid is water or the intestinal fluid, and m is the you can say the membrane, जो plasma membrane है, a is the area, d is the diffusion coefficient, and h is the thickness of the membrane. So here you can see that this is the concentration of drug inside the GIT, a is the area, जो area उसका वो है, greater is the area, greater with the absorption, and this is the thickness. Basically the lesser is the thick the membrane, more movement of drug will occur. तो जितनी ज़्यादा ये thick होगी उतनी drug की movement कम होती है, because drug has to cross the membrane, and if it will be thicker, it will be very difficult for the drug to cross that membrane. So DCB, rate of movement of drug inside the rate of change of drug inside the blood, I mean कि जब blood में drug concentration increase होती है with respect to time, so that way with the positive sign, D is the diffusion coefficient. Again this is the basically the km over f, that is the partition coefficient, and a is the area, h is the thickness, c is the concentration in the GIT, and cb is the concentration inside the blood. So that is almost all equal to this one, because cb is equal to around 0 from the when it is going to be absorbed, the process start hota hai, it is almost all equal to 0, so we can neglect the value of cb. So that's why Km over you can say the F, it is equal to partition coefficient. ये partition coefficient की equal है, and then again between the membrane and the fluid, in this case the aqueous fluid, is the surface area, greater is the surface area, and H is the thickness. So all these things, they represent that the rate of change of drug concentration inside the blood is directly proportional to this coefficient, diffusion coefficient, surface area, and it is irreversibly proportional to the thickness, and it is also directly proportional to the concentration of drug inside the GIT. Greater is the drug concentration in the GIT, greater will be the movement of drug from the GIT into the blood. So the again the partition coefficient, which is you can say the Km over F, this represents the concentration of drug in the upper phase, that is mostly the you can say the organic phase, and concentration of drug in the lower phase, that is mostly the aqueous phase. Lower means because the density of water is greater as compared to the organic phase, that's why it is called as the concentration of drug in the upper phase, and this is the concentration of drug in the lower phase. So that is the organic, means the जो हमारी लिपिड मेम्ब्रेन की कंपोजिशन है, दैट इज लिपिड bilayer, that's why we use the organic. And जो water है or the intestinal fluid है, that is mostly the fluid and aqueous fluid, so it is the aqueous. And you can use the oil, or you can simply you can say the this is the you can say the water. इसमें जो इसकी रेशियो एग्जिस्ट करती है ड्रग की, that is called as the partition coefficient Km over F. So that is basically about the you can say the partition coefficient का जो concept है, I mean Km over F, that is the partition coefficient. So based on this you can say that the if it is greater, which means that partition coefficient is greater, this means that the concentration of drug in the organic phase will be greater, so that means that there will be more transfer of drug from the GIT into the blood. Okay, so another thing is that if although we know that the movement of drug is always for the unionized form of drug, but if intrinsically the drug has low lipid solubility, then it will be very difficult for the drug to cross the membrane, although it will be available in unionized form. Okay, so keeping these things in mind, it is not only the unionized form of drug, but it is the physicochemical property that basically also has the effect on the transfer of drug from the site of administration to the blood. So next important factor is the pH partition theory of drug absorption, that basically link about the pH, pKa, and lipid solubility that affect the absorption, and this is called as pH partition theory of drug absorption. तो ये चीज़ें जो हैं लिपिड, अगेन जो लास्ट स्लाइड में मैंने आपको बताया, दिस इज नॉट ओनली द पीएच पीके but also the lipid solubility of drug also affect the transfer of drug or the absorption of drug. So drug की intrinsic जो property है, I mean कि उसकी lipid solubility, that also affect the transfer of drug from the higher concentration to the lower concentration. Okay, so according to this pH partition theory, the drug absorption is always through the passive transfer. Drug is basically absorbed in unionized form, and drug should be that drug has to be enough lipid soluble to क्रॉस द मेम्ब्रेन. तो ड्रग की लिपिड सॉल्युबिलिटी भी अपनी होनी चाहिए कि वो उस मेम्ब्रेन को क्रॉस कर सके. So this is all about the today's lecture, that basically about the Fick's law of you can say diffusion, pH partition theory, that basically affect the passive transfer of the drug from the GIT or from the site of absorption to the blood. So if you have any question, you can ask the question, and I will be pleased to answer those questions. Thank you.